When you look at a plant or a medicine, you're actually looking at a highly advanced, microscopic biochemical factory. Today, we aren't just talking about surface-level benefits. We're zooming all the way into the atomic level. We're going to map the chemical architecture of Azadirachta indica, commonly known as the Neem tree. Throughout this explainer, we'll decode its chemical structures and reveal exactly how these molecules interact with human cellular receptors to impact everything from metabolic control to completely novel nanotechnology. Welcome to the molecular blueprint of Azadirachta indica. Here's our roadmap for this explainer. We'll start with nature's biochemical factory, focus on limonoids as the master defenders, dive into metabolic control at cellular levels, and finally wrap up with green synthesis and nanotechnology. Let's get right into it. For thousands of years across Ayurvedic and Unani traditions, this species has been so revered, it's actually colloquially called the village pharmacy. Literally, almost every part of the tree gets utilized for its anti-malarial, antibacterial, and anti-inflammatory properties. But modern pharmacology asks a totally different question, right? How exactly does it do this? To answer that, we've got to look at what this biochemical factory is actually manufacturing. Through advanced analytical techniques, think high-performance liquid chromatography and mass spectrometry, researchers have isolated over 400 distinct bioactive compounds in just this single botanical source. It's a massive, multifaceted chemical diversity that, honestly, synthetic chemistry really struggles to mimic. Section 1. Nature's Biochemical Factory. Decoding the Arsenal. Let's categorize this foundational blueprint. The chemical arsenal here is primarily divided into major classes. First, we have limonoids, featuring complex molecules like azadirachtin that act as potent insecticidal agents. Second, flavonoids, like quercetin, which are basically your profound antioxidants, scavenging for free radicals. And third, terpenoids, such as pelargone, driving crucial biocontrol mechanisms. By tracking these specific classes, we can see exactly how they behave when introduced to human cellular pathways or even invasive pathogens. Section 2. Limonoids, the master defenders. These complex tetranort triterpenoids make up roughly a third of the plant's characterized constituents. They're essentially the plant's heavy artillery. But how are they actually built? Well, research using stable isotope labeling shows they're constructed primarily through the mevalonate or MVA pathway. You can think of it like the plant's internal cellular assembly line. In fact, when researchers introduced specific chemical inhibitors to just shut down this assembly line, limonoid levels tanked. This precise metabolic engineering is what allows the plant to synthesize incredibly intricate structures like nimbolide and azadirachtin. When we lock onto specific targets, the pharmacology gets absolutely fascinating. Take nimbolide, for example. It specifically modulates tumor-suppressing genes like P53 and P10, directly commanding tumor suppression and cellular apoptosis, programmed cell death. Then we have solanin, which is a potent anti-melanogenesis agent, and of course, azadirachtin, which physically interrupts insect reproduction. It's a world-class biopesticide. Look at their anti-inflammatory mechanics. When macrophage cells are induced with inflammatory toxins, specific limonoids don't just, mask the resulting inflammation. Actually, Western blot analysis proves they physically reduce the expression of COX-2 proteins. They're literally shutting down the enzymes driving the inflammation right at the genetic source in a concentration-dependent manner. And the absolute best part? They do it without harming healthy cells. Moving on to Section 3, metabolic control at cellular levels. Let's consider the natural molecule nimbidiol. It's a diterpenoid isolated right from the plant's roots. In bioassays, researchers put it head-to-head against standard synthetic diabetes medications, drugs like acarbose and voglibose. And the result? No way around it, nimbidiol was actually a more potent inhibitor of specific intestinal enzymes than those commercial synthetic drugs. Here's the exact pharmacological mechanism step by step. When nimbidiol enters the intestinal tract, it acts as a competitive inhibitor. It's basically a molecular roadblock. It binds to carbohydrase enzymes, totally blocking them from breaking down complex sugars. Because that carbohydrate absorption is significantly delayed, those rapid post-meal spikes in blood glucose and insulin are flattened out, safely guiding the body toward normoglycemia. But beyond just glucose control, these botanical extracts execute some really remarkable interventions across other biological systems. For instance, when dealing with malarial parasites, the extracts act by redox perturbation. That means they impose an extreme oxidative stress test that the organism simply cannot survive. And what about human organ protection?
Well, in the kidneys, methanolic extracts of neem actually protect mammalian tissue from highly toxic chemotherapy drugs like cisplatin by reversing lipid peroxidation and restoring cellular defenses.
And the quantitative data totally backs this up. When Azadirachta indica leaves were combined with other therapeutic plants, the resulting fractions demonstrated a staggering 91% chemosuppressive effect against malaria in vivo. That's approaching the 100% suppression you'd see with standard chemical interventions like chloroquine. And it's all driven by compounds acting as multiple inhibitors of crucial malarial proteins. Finally, Section 4, Green Synthesis and Nanotechnology. You see, our utilization of plants is really evolving. We no longer just extract chemicals to consume them, we're now using the plant's own molecules to engineer completely new materials. Green synthesis utilizes the chemical groups in neem to act as natural stabilizing agents. So instead of using highly toxic industrial chemicals to fabricate metal nanoparticles, scientists can just use a simple eco-friendly plant broth. It's a real game changer. This creates entirely new weapons. Neem-synthesized silver nanoparticles are out there destroying fungal biofilms that have actually grown resistant to standard drugs. Zinc oxide nanoparticles are halting agricultural pathogens. And in the realm of environmental cleanup, these magnetite iron oxide nanocomposites can degrade over 70% of toxic industrial dyes right out of the water. Check this out. When zinc oxide nanoparticles are synthesized with neem extracts, the plant proteins literally shrink the particles and increase their specific surface area. This results in a staggering 72% response rate when they're used as ethanol vapor gas sensors. That's absolutely crucial for things like breathalyzers or industrial safety monitoring. And the kicker? They operate at much lower temperatures. It's a perfect marriage of botanical biochemistry and advanced material science. I want to leave you with this final provocative thought. We've spent this explainer unpacking over 400 compounds, mapping mevalonate pathways, tracking competitive enzyme inhibition, and synthesizing metal nanoparticles from just a single species. So if one tree holds this staggering level of complex molecular architecture, what other clinical blueprints are hiding in nature's pharmacy just waiting to be decoded? The molecular frontier of natural products isn't a thing of the past. As we've seen today, it is the absolute cutting edge of our scientific future.